Hello everyone. Today we explore a very important and fascinating topic in microbiology, the normal flora of the human body and why it matters. At first glance, you might think microorganisms are always harmful, but many are harmless and some are essential partners for human health throughout life. We will explain where microbes live on and in our bodies, how they colonize, what roles they play, and when colonization begins. This 5-minute explainer will build step by step from sterile internal tissues to the diverse communities that live on our external surfaces. To begin with, in a healthy human being, certain internal tissues such as blood, brain, and muscles are normally free of microorganisms. These internal sites remain sterile thanks to physical barriers, tightly regulated blood flow, and robust immune surveillance that prevents microbial colonization under healthy conditions. External surfaces tell a different story. Skin and mucous membranes are constantly exposed to the environment and readily acquire microbes from contact and air. Microbes that establish themselves on these surfaces form stable communities influenced by local temperature, moisture, pH, and available nutrients. This collection of microorganisms that regularly inhabit a particular site in the body is known as normal flora or resident microbiota. Normal flora includes bacteria, fungi, archaea, and in some contexts, viruses and protozoa, each adapted to specific niches on the host. An interesting point to note is that a fetus inside the uterus is considered sterile in most classic models of human development. So when does colonization begin? Colonization typically starts at birth, when newborns encounter microbes during delivery and immediate postnatal contact. Vaginal delivery exposes newborns to maternal vaginal and gut microbes, while cesarean delivery often results in initial colonization by skin and environmental organisms. After birth, breastfeeding, skin-to-skin contact, family members, and the home environment rapidly shape a newborn's early microbiome within hours and days. Over weeks and months, diet, antibiotics, hygiene, and other factors influence which microbes persist and how the microbial community matures. Early microbial exposures help train the infant immune system, support digestion, and offer protection against some pathogens during a critical developmental window. Skin hosts microbes adapted to dryness, salt, and fatty acids. These organisms often localize in hair follicles, sebaceous glands, and moist creases. Mucous membranes lining the mouth, nose, throat, and digestive tract support dense, diverse communities that interact closely with host tissues and immune cells. The gut microbiome is the largest and most metabolically active, breaking down complex carbohydrates, synthesizing vitamins, and producing signaling molecules. Oral microbes assist early digestion, influence breath chemistry, and can impact systemic health when balance is disrupted by disease or poor hygiene. The urogenital tract supports location-specific communities that play roles in reproductive health and resistance to opportunistic infections when balanced properly. Across these habitats, resident microbes compete with pathogens, modulate inflammation, and produce beneficial metabolites like short-chain fatty acids important for health. Maintaining a balanced relationship with normal flora is essential. Disturbances can allow opportunistic organisms to overgrow and cause local or systemic disease. Common disruptors include broad-spectrum antibiotics, dietary changes, stress, illness, hormonal shifts, and altered hygiene or environmental exposures. To support healthy flora, prioritize appropriate antibiotic stewardship, a diverse fiber-rich diet, measured probiotic use when supported by evidence, and gentle skin care. Researchers now use sequencing and computational tools to map microbial communities and identify links between microbiome patterns and health outcomes. Therapies such as fecal microbiota transplantation or targeted bacteria therapy aim to restore healthy communities in specific clinical contexts under medical supervision. In summary, most microbes that live on and in us are beneficial partners, crucial to development, digestion, immunity, and overall human health. Thank you for watching. And that wraps up today's topic. If this explanation helped you understand the concept more clearly, make sure to like the video and subscribe to this channel for more simplified lab technology and radiology lessons powered by AI. we break down complex subjects in hematology, biochemistry, microbiology, pathology, X-ray, MRI, and diagnostic imaging, step by step, so you don't just memorize, you truly understand. If you have any questions or want a specific topic covered, drop it in the comments below. Until the next video, keep learning smart, keep thinking critically, and keep mastering diagnostics.